My name is Kelly Madsen. I'm the Senior Director with the Healthcare Partnerships team at the Muscular Dystrophy Association. And on behalf of MDA, I want to thank you for all you do for MDA and the neuromuscular community. MDA's industry update webinars are programs which provide an opportunity for the medical community to receive information and updates impacting care and treatment directly from pharmaceutical and biotechnology companies. These webinars are just a part of MDA's medical education resources, and we encourage you to visit the medical education pages on mda.org. Regarding this webinar today, if you have any questions that you'd like to pose regarding the webinar, you can put them in the Q&A. And now I'd like to introduce our topic for today's webinar. Edgewise Therapeutics is currently investigating a novel therapy for Becker and Duchenne muscular dystrophy, two very serious and debilitating muscle diseases. Today, the Edgewise team is here to share the latest updates with their Becker and Duchenne clinical programs. I'd like to introduce our presenters in today's webinar, John Ling, the Executive Director of Medical Affairs at Edgewise Therapeutics, and Joanne Donovan, the Chief Medical Officer of Edgewise Therapeutics. I will now turn it over to the Edgewise team.
Hi, Kelly, and hello, everyone. Thank you for inviting us to present during this MDA Industry Update webinar, and thank you to everyone for joining and for all the work you do within the community. I'll do a quick overview of our programs and who we are as a company, and then I will hand off to Joanne, and she'll do our Becker clinical program update, and she'll talk about our Canyon and Mesa studies. And then she'll then go on to talk about the Duchenne clinical program update and talk about Lynx and Fox. And we'll hopefully have time for questions and discussion at the end. So next slide, please. Thank you. So Edgewise, I really enjoy working with this company. We are leaders in muscle disease, and we have a focus on muscle science. And we have a rapidly advancing portfolio of small molecule development. So it's a combination of taking what we know about the physiology and science behind muscles and how they contract, and then small molecule development. And so we're advancing two programs. Sevasemten is an investigational drug in muscular dystrophies, including Becker and Duchenne muscular dystrophy. And then EDG-7500 is an investigational drug in obstructive and non-obstructive hypertrophic cardiomyopathy. And then we have some novel cardiometabolic targets. So we basically have some really deep programs of both skeletal muscle and cardiac muscle. Today, we'll primarily talk about the skeletal program in sevasemten. But importantly, it's unwavering patient commitment. And I just want to take a moment to thank all of the patient community for everything that you do in participating and joining in clinical trials, natural history studies, and just really activating your community. And as well to thank all the HCPs and other staff that are involved in running the clinical trials. It really does truly take a village to generate this data that you'll see in part today. Next slide, please. So our sevasemten clinical program, we have completed ongoing clinical trials. I've been at the company for 3 years, and I'm really proud to see the advancement of our programs over the past couple of years. We have ARCH, our phase 1 Becker study, which has been completed, and we've been presenting data over the past couple of years for those men that joined the ARCH study. And then we have DUNE, which is a phase 2 Becker, a phase 2 study that's an investigational exercise science study in Becker, limb-girdle, and McArdle. And then most recently, last December, we showed data from a phase 2 Becker program called Canyon. And then a pivotal cohort of that trial is called Grand Canyon. And that is a large global study that is active and not recruiting while all the other studies are completed. And then, as you see here, MESA is our Becker open label extension study, which is enrolling by invitation, which means that all the men or boys that were in our Becker studies can roll over to MESA and continue to collect data and stay on treatment. And then for Duchenne, we have Lynx, which is a phase 2 dose ranging study, and we had presented some data back in June, which you'll see some more of that today. And then Fox, which is a phase 2 study of boys in Duchenne who are currently on gene therapy treatment, looking at the safety and pharmacokinetics of sevasemten in a gene therapy treated background. Next slide, please. So a quick overview of Becker muscular dystrophy clinical program update. Next slide. So Becker is a serious, progressive, debilitating neuromuscular disease. And I'm really proud of the community over the past couple of years at how much they have become activated, where men and boys with Becker are starting to find each other and tell their stories. It's an X-linked recessive disease caused by mutation in the DMD gene, but this is an in-frame mutation that, although it produces a smaller dystrophin, it's not a fully functional dystrophin. You'll see the impact of that in a moment. It's a multisystemic disease that can lead to irreversible loss of muscle function and premature death. And many men with Becker have cardiac issues as well. There are currently no treatments that are approved for Becker, and it's really just through disease management and physical therapy. Next slide. So in dystrophinopathies, fast muscle fibers are disproportionately injured by contraction. I mentioned that, Edgewise being deep in the muscle science. When you go back and look at some of the early data, it has showed that these muscle fibers are preferentially damaged, and we're able to develop a small molecule, sevasemten, which binds specifically to the myosin molecule of these fast muscle fibers. So what happens during normal, healthy muscle contraction, and you see on the left here, that when an individual muscle fiber contracts, it's not doing it alone. When you have a proper level, amount, and function of dystrophin, as well as other molecules, then that individual muscle fiber can contract and share that load, that contraction-induced stress with its neighbors. So that contraction-induced muscle injury is part of normal muscle use and development, but healthy tissue can repair that damage. Whereas in dystrophic tissue, that damage happens in a cycle over and over and over again. So as you see on the right now, without a proper level of dystrophin, whether a full-length dystrophin or no dystrophin at all, you see that that muscle fiber in the middle now is contracting by itself. And when it does that, all that stress and that load is put onto that muscle fiber, and you get breakdown of the plasma membrane, leakage of the calcium, proteolytic pathways, activate, and that muscle fiber breaks down. And that cycle then will repeat itself over and over again so that the tissue can no longer repair itself, and you get replacement of that muscle fiber with fat and fibrosis. And so the contraction-induced muscle injury is the root driver of the disease pathophysiology. In the next slide, you'll see this in action. So as I mentioned, that fast muscle fibers are preferentially damaged. We developed sevasemten to target fast myosin. And this is a preclinical model, I must stress, that we're looking at the limbrical muscle of a mouse. It's a finger muscle of the mouse. And on the left, you can see that when this muscle fiber, it's sped up, and we can see the contraction. And under that contractile stress with no dystrophin, because the index muscle lacks dystrophin, you see the muscle fibers actually breaking down, and those little blebs or little balls that are forming, that's the broken down muscle fiber. On the right, when you treat with sevasemten, what we're trying to do is, by targeting that fast myosin, the ATPase domain, we're slowing down contraction. In this example, we're contracting at about 85% in the presence of sevasemten. So by slowing down contraction, that muscle fiber can contract and not have the same damage. Now, this is a preclinical mouse model, but what it did is it gave us confidence that as we move into human trials, that we understand and see what our mechanism of action looks like. So next slide, please. So as we move into humans and we think about clinical endpoints, the NSAA is a well-established and validated measure of global function that is clinically meaningful in a real-world context. So we're asking the men or boys with Becker to do the NSAA, which is a 17-item test where you hop on left leg, hop on right leg, stand on heels, and so on and so forth. And you get points for whether or not you can do that. 2 points for performing it normally, 1 point for doing it with compensation. And if you can't do it, you get 0. And so we often think then, what does the NSAA mean in real life, right? Jumping and hopping, the activity that you associate that with is playing sports. Other things like walking on uneven ground, getting up after a fall, independent outdoor mobility. So these functions that we're looking at have real world context. So you think about, what does 1 point loss on the NSAA mean to a man or a boy living with Becker muscular dystrophy? So the next slide, please. So over the past few years then, there have been multiple studies coming out looking at the natural history of Becker that supports functional decline measured by NSAA, and that it's consistent and predictable. So as mentioned, there's 17 items you can do. So you can have a score of 34. So it turns out that some men with Becker when they're at 33 or 34, they could be considered a slow progressor and they float at that top of the NSAA scale, and that's somewhat of a ceiling effect. But the data will show you that once you get below 32 in the NSAA score, there is a continuous and relentless decline in the NSAA. So in the studies I've looked at the NSAA function, they've looked at and included patients above 33, above 32 and below 32. But even with that mixture of data, you see that is a 0.9 to 1.7 point rate of decline on the NSAA, and that is relentless and irreversible. So we can go back to the previous slide and think about what does that mean in daily life? They're losing those functions over time as they're living their life. So next slide, please. So at that point, then we want to roll it over to Joanne for the Canyon and Becker clinical updates to show you what this data looks like in real life. Thank you.
Thanks, John. So I'm delighted to speak with you all today. Those of you who are out in the field and treating these patients on an everyday basis. So within our Canyon study, this was our first phase 2 randomized placebo-controlled study to look at sevasemten's safety and its effect on biomarkers. Now, the primary endpoint of this study was biomarkers, the change from baseline in CK averaged over the last 6 months of the study. And we looked at men and adolescents aged 12 to 50 who had a dystrophy mutation and a Becker phenotype, so they were walking after age 16, not on corticosteroids. And to what John showed, they had a North Star between 5 and 32. So they were in that group that was relatively rapidly declining. And once they start to decline, as John said, they tend to continue to decline. So we enrolled 40 adults, 29 adolescents, and treated them for a year with study drug or with placebo. The adolescents, we did more of a dose ranging study, two different doses. And we looked at functional measures, including the North Star, as well as safety, PK, biomarkers, and other functional tests. And on the next slide, what we saw from a safety perspective, now that's the first thing we're looking at in this longer term study with men. And treatment emergent side effects that occurred in more than 5% of patients are shown here. The most common were headache and dizziness, and they tended to be transient and only in the first few days of dosing. I should add, this is a once a day tablet that is given, typically we give it at night. On the next slide shows what we saw in terms of the biomarkers. Now, when you picture those mouse lumbrical fibers causing with damage, what of course they do is leak and they show CK. So that's what often brings people to see neurologists. They have an elevated CK and brings them to medical attention. Well, we saw the CK, which is reflective of damage to the muscle fiber, go down 28%, statistically significant. And importantly, what's called TNNI2, which is the troponin that is specific to the fast muscle fibers. And that went down by 77% compared to placebo, highly statistically significant. So that tells us that the drug is getting to muscle and it is protecting against contraction-induced muscle damage, the kind that happens with everyday activities. So what's happening in terms of function? Now, the study wasn't powered for North Star. This was our first study. And on the next slide, you can see that these men had, and actually they had an average North Star of about 15. So they were fairly advanced in their disease. And what we saw in terms of the change over time, the North Star, was basically in the folks that were treated, the adults here that are treated with sevasemten, it was stable over time. Now, this is a year. You're looking at a disease that's going on for many years. And so that's a snapshot. That's good. What we did also see was that the folks that were given placebo declined consistent with the natural history studies. So it was about 1.1 points per year. Now, this was not statistically significant. The p-value was 0.16, but it tells us we're moving in the right direction. And again, it was well tolerated in this study. On the next slide, this was presented at MDA in March. So to summarize, the Canyon part of the study is well tolerated, no safety concerns identified, no liver toxicity, no cardiac toxicity, no renal toxicity. We hit the primary endpoint, the first study in Becker to do so. And we also hit a more specific biomarker, the plasma fast skeletal muscle troponin. We saw that patients were stabilized. And when we've talked to patients, when we've done surveys, stabilizing disease, you've lost muscle. You're not going to build back the muscle, but stabilizing disease and keeping things where they are is incredibly important to these patients. And actually, there was improvement compared to the placebo group, which declined in line with the natural history. I didn't show you, but other functional endpoints trended favorably. Now, we are trying to see, yes, the next slide. We looked at patients, and the next slide, we brought these patients all into an open label study called MESA. I will add, these are all rock formations. That's our theme. Our company is based in Boulder, Colorado. So these are all Colorado outdoor themes. And we included in this open label study men in all of the studies to date. So that gives us additional information. It allows us to follow them, get additional safety data, continue to follow the biomarkers over time, as well as function. And the last time we did a data cut, 99% of the eligible men had continued on into the open label study, which is encouraging to see that happening. As it was mentioned, we are also currently doing the Grand Canyon study, an 18-month pivotal cohort. That's fully enrolled. We're not recruiting at that point. Those patients, as they finish, are also moving into the MESA study. On the next slide is what we saw in terms of the function. Now, you saw the part on the left here, up to 12 months. That's what happened in Canyon. Now, what happens when they're treated for an additional 6 months? And they continue to be stable. And in fact, the placebo men who went down over time are now stable as well. So this is encouraging we go out longer. And also, this is the time period that we're looking at in the pivotal study. Now, these patients are remaining blinded. That's important for the FDA, that these patients continue to be blinded to their initial treatment assignment. So this isn't a reaction to, oh, now I'm on active drug. Also, we had no new safety concerns. Now, this is a rare disease, about 5,000 patients in the United States. But what we want to do is get every piece of information we can from this study. And on the next slide shows the MESA data, the open label data. And what we did was we developed, in conjunction with folks at the University of Leiden, Professor Eric Nix, to develop a predictive model to say, okay, if you are this old, you have this North Star, this function, what is predicted to happen over time? And looked at different factors. This model accounted for a little over half of the predicted change in North Star. So then we can look to see, are the patients in the study, were we just lucky that the patients who were on drug were predicted to do well, and the patients with placebo were the ones that were worse off? And so we can look at what happened in the trial and compare it to the natural history, as shown on the next slide. And what we saw was that you saw the top part, 18 months of treatment. And based on those individual patient baseline characteristics, they would have been predicted to go down by over 2 points. So that's quite encouraging. And when you think back again to what John was showing, these are, each point is either a compensation or a loss of activities like climbing up a curb, being able to get up off the ground, being able to step up or down. So these are important things. And this is presumably a year after year progression of symptoms, as you saw in the natural history studies. Now, we had an earlier study, ARCH, in which 12 men were treated in an open label study for now up to 3 years. And on the top, you can see that over now 3 years, we have 11 of the 12 that are continuing to be treated. And they are stable over 3 years. And the longer we go, the more encouraging it is to see that they are stable over time. And so you're really greatly reducing the disease progression is what it appears from this, admittedly, this open label data that we're looking at. And again, we went back to see what would they have, what would the model predict would have happened to these men. And they would have gone down by about 4 points. So again, this is encouraging. Now we have almost 40 men treated out at 18 months to 3 years. So we are looking forward to the data from Grand Canyon, and we anticipate that that will support, if positive, that will support a filing for an NDA, and that those data will be out towards the end of next year. Now, as was mentioned, we're also looking at Duchenne. And you are well aware in the next slide of the continued need, really, in Duchenne. And we're looking to develop a mutation-agnostic mechanism of action that could be used as a foundational therapy, either alone or combination with other treatments. And again, we're looking at contraction-induced injury, so it's complementary to other treatments. On the next, we've had 2 studies, the LINK study. These are cute little animals that are around in Colorado. And that is a study in boys that are between 4 and 9. The FOX study looked at sevasemten on top of previous gene therapy and children and adolescents that were previously treated. Both of these studies are fully enrolled. And their goal was to determine a range of doses to identify a potentially beneficial dose for a phase 3 study, look at the patient population to design that study. And it was a 3-month placebo-controlled study followed by an open label period. And these patients were progressively transitioned to higher doses. And what we saw on the next slide in terms of safety, we looked at a number of doses, and as you see, there are higher doses even than we looked at in Becker. And what we looked at similar endpoints in terms of North Star and other functional measures. And on the next slide, what we saw in terms of safety was it was pretty well tolerated in this in the 3-month placebo controlled period. And similar in the open label. What we did was we identified a target dose of about 10 milligrams. This is a mechanism in which you don't necessarily, more may not be more. So we went up, we backed down to a 10 milligram dose. And after now the boys on the next slide are out, the boys that were dosed at about 10 milligrams over a period of up to about 18 months. This is now not in the placebo-controlled part of the study. And we compared them to a multivariate regression model that was published in the literature, which again, based on individual baseline measurements. North Star looked like it was better, about 2 points than would have been predicted. And we also looked at a measure called SV95, which is the maximum stride velocity. It is a new measure that is very well validated and correlates with other functional measures, and is also reflects about 3 weeks in everyday life. And that was the progression, or the advancement in terms of it would typically go down with time. It was almost stable. So that was very much different than you would have expected with natural history. So again, this is not a placebo-controlled study, not a study that we're ready to say, yep, we have something for sure, but it is encouraging, and it does help us identify a dose. We also looked in Fox in boys who were on gene therapy, and about 2/3 of them were on Elevidys before. In terms of safety, in this group, we did see that at the next slide, on the higher doses, we started to see the boys slowing down, basically. So we backed them down to a 10 milligram dose, and that was well tolerated. So to summarize, we have functional observations that support the 10 milligram dose. It was well tolerated, no new safety concerns, and we are continuing in the open label part of the study to collect longer term data. And we're going to the FDA to get input on the phase 3 design of these. And so we are excited about the prospect in Duchenne as well. So in closing, thank you, first of all, for joining today. Very much appreciate the contributions of the patients, the investigators, the study site personnel, and everybody who has participated. I would like to give a shout out to Becker Education and Engagement Day that's happening next month at multiple sites across the country. It's been very well received and is growing. This is the 3rd time that it has happened. And here is the link to encourage your patients or their families to register. Really, there is support for travel for them. And people have found it very helpful in the past to hear from experts and to meet others in the community as well. So we're looking forward to data in Becker next year. We'll be back with more information. And we're happy to entertain questions from people. I am looking at I'm looking at it. I don't see questions. One frequent question I get is, what about the cardiac effects? And because we mentioned troponin, there are different troponins, and there are actually different myosins in cardiac muscle as well compared with fast skeletal muscle. So this drug does not affect cardiac troponin, doesn't affect cardiac contraction, and we didn't see any change in echoes. And it also doesn't affect smooth muscle, so other places where troponin, where myosin is present as a contractile protein. So that's a frequent question. We are not recruiting at this time, which is, we are in the home stretch kind of in terms of the clinical trial. Anything else out there? Otherwise, it's a minute from the end, and we can thank you for your attention, and hope that this has been useful to you to hear a little bit more about Becker and about sevasemten, and hopefully its role in helping people in the future.
Great. Thank you so much to Jillian and John, and thank you to the medical community for attending today. And we hope that you can join again. And thank you for the very insightful information to the Edgewise team. Appreciate your time today. Thank you, everyone.
Thank you.
Bye-bye. Thanks.
Bye.